Our next speaker is Diego Giraldo from Johns Hopkins. Diego, do you want to start sharing your screen? And while Diego is getting set up, just a little bit about him. So Diego obtained his undergraduate degree in biology from Universidad de los Andes in Colombia. He then moved to Germany and did an MSc and PhD in neurosciences at the University of Göttingen, where he studied molecular mechanisms of Drosophila mechanosensation. Diego is currently a postdoc in the McMenamin Lab here at the Johns Hopkins Malaria Research Institute, and he is currently studying the neurobiology of mosquito attraction to humans. So take it away, Diego.
Thank you, Tori. Am I on the correct slide or are you seeing presenter mode?
Nope, you are good.
I'm good. Okay. Thank you for the introduction and thank you for the invitation to present my work today. I work at the McMenamin Lab at the Johns Hopkins Research Institute and the School of Public Health. And today I'm going to talk about our semi-field system for tracking Anopheles gambiae olfactory preferences. So as we just heard, mosquitoes use different cues to find and bite humans. And in the long range, we have CO2 and body odors. And as we come closer, we have body heat and moisture, as we heard. And then we have taste hints that lead to biting and disease transmission. We focus in long range cues such as CO2 and body odors. We know that human odor is a very complex mixture of compounds. And there's hundreds of components that lead to lots of variation among people. And also it varies between different body regions of a person and it can be influenced by the microbiome, hygiene practices, diets. So as I mentioned, we can have different people that have different odors, and this can lead to differences in preferences of mosquitoes and mosquitoes biting more often or being more attracted to some people than others. And studying this sense of smell of a mosquito can help us develop strategies of control, whether it's by developing repellents that inhibit this host detection or generate repulsion, but also creating chemical lures that can attract mosquitoes and can mimic this highly attractive human odor. And these preferences are not just annoying for people that are very attractive to mosquitoes, but they can have an epidemiological impact. So in this specific study, they looked at a population that was at risk of being bitten by mosquitoes. And what they found was that a very small proportion, so only 20% of the population accounted for 80% of the blood that was matched in mosquitoes, meaning that mosquitoes were only biting a very small proportion of this population. And this increased biting due to these preferences can have an important epidemiological impact. So we have different ways of measuring chemotaxis and these attraction to odors in the lab, which go from uniport olfactometers, where mosquitoes go through sort of a tube to an odor that's attractive, dual port olfactometers, where mosquitoes are presented with different stimuli, and slightly bigger ones, such as wind tunnels, where they're a few meters long and mosquitoes can fly upwind to an odor. But even though they're very important and we can get a lot of information from them, there are limitations, basically because conditions are very controlled and the size of these setups cannot be too large because they're in labs. So we want to study whole human odor and preferences to whole human odor. What we want to do is we want to study this in a much natural or semi-natural context. So we did, what we want to do is determine these olfactory preferences of Anopheles gambiae specifically in Zambia. And what we did was we built a large scale assay to assess the preferences. And by this, we built this very large semi-field cage. And the goal is for it to allow us to compare attractiveness of odor from multiple subjects in a large flight arena and identify individuals that are very attractive and try to find chemical correlates of this attractiveness. So basically trying to understand what makes humans very attractive. What I'm going to talk about today was the first stage of this study, which is we first wanted to test the cage and determine whether Anopheles gambiae females could detect olfactory stimuli that are brought into the cage and also detect thermal cues that we have in there. We want to know whether they could detect human odor, and more importantly, we wanted to see if the females would show olfactory preference between two humans and if they would consistently prefer one human over another. And all the work I'm going to present today, I collected all the data in collaboration or with the help of Dwayne Jackson, a grad student in the lab. And this was all done at the Macha Research Trust in southern Zambia, and the collaboration of the staff over there. This is a schematic of our cage. So it's a 20 by 20 meter cage. And surrounding the cage, we have tents where humans can sleep inside. And in front of each tent, we have a fan that brings all the air, all the odors inside of the tent into the cage through some ducting. And at the end of the cage, we have these heated platforms that will mimic human skin. So when mosquitoes detect an olfactory cue, we expect them to land on these platforms. Here I'm showing you some drone footage of the area. So again, we have our large flat cage where we can release mosquitoes and let them fly around, and they're exposed to natural elements. And here we have the tents surrounding the cage with the ducting that connects them to the cage. And again, we have this 20 by 20 meter cage. The tents are 15 meters away, so the mosquitoes are really only getting whatever odor, whatever air we're bringing inside, and they cannot fly up the ducting, so they're just getting the odor inside of the cage. And we have a weather station that was constantly collecting weather data. This is a view of the cage inside, and at the end of each ducting, we see what we'll call these odor-guided chemotaxis assays. And this basically consists of the ducting that brings in the odor, the heated platform that has some infrared light so we can see the mosquitoes landing, and a camera on top that is recording the activity of the mosquitoes as they land on the platform. And we can measure how many mosquitoes landed and the occupancy of these platforms and use that as a measure of attractiveness. And here I'm just showing you a video of mosquitoes landing on these platforms. So the first question we had was whether mosquitoes could detect an important olfactory cue such as CO2 and land on a platform. So again, we have these small platforms that are 10 centimeters in diameter and this large cage where mosquitoes are flying around. So we wanted to know if mosquitoes could detect the relevant odor stimulus and land on the heated platform. So we had one tent that was baited with CO2, and the rest of the tents were left empty, so they just had clean environmental air. Every night we would rotate the position of the CO2 randomly, and this was to account for any potential biases of position within the cage. And we did this over 6 nights. And the way I present the data is CO2 is platform number 1, and then the control platforms increase in a clockwise manner. Here I'm showing the occupancy of the platforms in one example night. So as we can see, the CO2 baited platform had activity and had landings of mosquitoes throughout the night, but the control platforms were left empty. So mosquitoes were not attracted to just heat alone, and only in presence of CO2 would they land on the platform. If I quantify this over the 6 nights, and I show the mean of landings on the target throughout the night, so we record for 6 hours, we can see that mosquitoes would land on the CO2 platform, but they were not attracted at all to the control platforms. So this tells us that mosquitoes could detect an olfactory stimulus and be attracted to it, and that heat alone with just air would not create any landings or lead to any landings. The next question we had was whether human odor would be preferred over CO2 and whether mosquitoes could detect this more complex combination of odors. So as in the previous experiments, we had one tent, in this case, baited with human odor, so humans sleeping inside the tent, and the other tents were baited with CO2, and the position of the human was rotated every night, again in a random manner, and this was done over 6 nights. And we look at the occupancy of the platforms, we can see that in the CO2 platforms, we still get some landings. It is still an attractive stimulus, and mosquitoes would be attracted to the heat and presence of CO2. But whenever we had a human sleeping in a platform, the attraction or the occupancy of this platform was much higher. And when quantified over 6 nights, we can see that, again, the human baited platform was very attractive. And throughout the entire night, we consistently had landings. And we had much fewer landings in the CO2 treatments, showing that mosquitoes can detect human odor in our cage, and they will prefer it over CO2. Now, the third and most important question we had was, is there preference between 2 humans? So if there's odor from different humans coming into the cage, will mosquitoes consistently prefer one over the other? So in this case, we had one human sleeping in a tent, another human sleeping in another tent, and we would rotate the position of human one throughout the night, again, to control for position effects. And then every night, we would position human two at a different position relative to human one, so that we could have all the possible combinations, meaning one night human two would be to the left of human two would be to the left of human one, but another night it would be across the platform until we had all 7 combinations. So we did this over 7 nights. And we look at the occupancy, we can see again, our control platforms did not have any landings. So heat alone does not trigger landings. But whenever we have human odor, we get a lot of landings, a lot of occupancy in these platforms. And in this specific case, we can see that it's much higher for human 2 than human 1. So when we do this over 7 nights, again, nothing in the controls, and we do see a lot of landings in both humans, but we see significantly higher activity in the human 2 platform. And this was consistent throughout the entire night. So throughout the 6 hours of the experiments, human 2 had more landings than human 1. If we look at the percentage of landings for every specific night, again, every single night, more landings for human 1. And we calculate a preference index where 1 would be preference for human 1, and -1 would be preference for human 2, and 0 would be no preference between the humans. We see that mosquitoes had a very strong preference towards human 2. So to summarize, we developed a method to study olfactory preferences of Anopheles gambiae in semi-natural conditions. And we found that mosquitoes can detect olfactory and thermal stimuli and land on our platforms. We found that heat and CO2 can trigger landing in our context, and that mosquitoes can detect human odor and prefer it over CO2. But more importantly, we found a setup that can help us screen for mosquito preferences between humans. Now that we know that mosquitoes, we can find preference between 2 humans, our next step is to see what happens if we test 8 people at once. So we have these 8 tents around the cage where we can potentially test 8 people at once. And we want to know if mosquitoes will consistently prefer the same people every night. And we can find very attractive humans in this context. And we then want to capture their odor and quantify it with GCMS to identify attractive compounds and try to understand what are these compounds that are making these people more attractive. And then try to use that to develop chemical lures that would help us improve methods of vector control. And one of the things that we want to do moving forward is that collaboration that we have with Dr. Bart Gurton at the University of Göttingen is using machine learning and neural networks for tracking. And this is just a quick example of just a network identifying our mosquitoes landing on our platform, which will help us analyze data much faster in the future. And again, be able to do a large screen of multiple humans in the cage or in the tents and try to identify attractive humans. Now, I would like to thank Connor McMenamin, our PI, for having great ideas and being very supportive. Again, all the data I showed was collected with Dorian Jackson. I'd like to thank everyone in the McMenamin lab and the Matcha Research Trust, especially Louis Kabinga, who was very helpful collecting these experiments. And I would like to thank you for your attention.
Thank you so much, Diego. That was super interesting, and you have a ton of questions. So the first one comes from Benjamin. Were the odor plumes turbulent or homogeneous?
So we have the odors coming in from the 10, 15 meters away, going through a fan, through inducting, coming out of this ducting in. So we didn't quantify this, but we would expect them to be turbulent within the cage once they come out of the ducting.
Okay. And then, so can you talk a little bit about how you can control CO2 coming from the person? Do you find different people put out different amounts of CO2?
Yeah, that's an interesting question. We do expect, and we have done some quantification in the lab. Stephanie Rankin Turner, who joined the lab last year, she's been doing a lot of experiments with this, and we do know different people do give off different concentrate or amounts of CO2. There's nothing we can control for. So the whole idea of this experiment is try to control as least as possible. Just have a whole human and try to capture everything that's coming off of this human in terms of volatile odors, but also including CO2, and bring it into the cage and try to find out who's more attractive and why. In the future, we do plan to quantify CO2 through the ducting. So potentially try to quantify and compare how much CO2 is giving off between participants.
Okay, and then kind of in a similar vein, do things like food habits, cleanliness, or even like race, ethnicity, would that contribute to potential differences in attractiveness?
Yes, I mean, these are all things that we think and people have tested that can contribute to these differences. In this specific context, the two people participating in the experiment had almost the exact same diet throughout a full month. They would shower 2 hours before the experiment with with shampoo and soap that gave off no scent. So we did try to control for that as much as we could. Of course, you would still get individual differences, and we expect all of these differences to be guiding how you smell.
Got it. And then one more question coming from Chris Potter. Really great work, Diego. Do you think the field assay might bias more towards volatile or abundant odors from humans and might not be sampling shorter range odors? How does the field assay compare to short range assays with the same humans, like the 2-port assay you mentioned?
That is a great question, Chris. Yes, we do expect that more volatile odors are more likely to go through the ducting. And as I mentioned, we do plan to sort of capture the odor and try to understand what are we bringing into the cage. We have not done, and it is something that we want to do soon, we have not tested preference between these two humans. Again, one of the there would be different things we could do, like we can have the humans, so the participants wear a nylon mesh on their arms to try to capture that more sort of closer range odor. So yes, this is something that would be interesting to do and try to understand how this field assay compares to, comparing the same people in the lab in a smaller setting.